Today's train destination, cure ALL, or Acute Lymphoblastic Leukemia. So asparagine is one of our protein letters, our amino acids. And it turns out that in some cancers, like this ALL, the cancer becomes really bad at making this. They don't make enough asparagine synthetase, which is what we use to make more asparagine. So they can only use asparagine that we get from like our food and that sort of thing, or from breaking down proteins. Our healthy cells can make our own asparagine. And we can also break down asparagine using asparaginase. And so our body makes this asparaginase so that it can regulate the levels of asparagine. But it turns out you can add extra asparaginase that you like purify from like bacterial cells, and you can add this. What happens if you add this to healthy cells is, well, they can compensate because they can make their own. But if you add it to these cancer cells, then because they can't make their own, they won't have enough asparagine to do all that they want to do, and so they won't be able to make all the proteins they need, and they won't be able to do all sorts of stuff, and so then they'll die. And so that is the rationality for the treatment of ALL with asparaginase. And so now let's talk about how how asparagine synthetase and asparaginase work, as well as some more about asparagine. And don't worry, it's not what makes your pee smell if you eat asparagus. The name does come from being discovered in asparagus, but it's not the pee smelly thing. Okay, so let's dive in. I am not a medical doctor. I inject proteins into Acto machines and nuts things into people. So please don't try to ask me medical questions. I can't give medical advice. I just thought that the biochemistry behind all this was cool and thought other people might be interested as well. We're talking about treating this disease called ALL, or Acute Lymphoblastic Leukemia. And so basically leukemia is like an umbrella term that refers to cancers that affect the blood or bone marrow, which is where the blood cells are made. So there's different types of leukemia that affect different types of blood cells. ALL is a type that affects these cells called lymphoid progenitor cells. So they're a type of white blood cell that's important for the immune system. And so this cancer can cause problems including anemia, fatigue, and immune system dysfunction. And so this treatment that we're talking about, this asparaginase treatment, more on the history in a bit, but it was first tried out in the 1960s. And now it's like a mainstay of treatment for childhood ALL, in addition to traditional chemotherapy drugs. So here's a basic overview, and then we'll get into the mechanism of how these enzymes or these reaction speeder uppers are working. So there's two main enzymes that we're going to be talking about: asparagine synthetase, or ASNS, and asparaginase, ASNase. And so basically asparagine synthetase is going to be our maker, and asparaginase is going to be our breaker. And so cells can make asparagine from the amino acid aspartate and glutamine. So basically take glutamine, take its nitrogen group, and stick it onto aspartate. So replace this oxygen. So basically you're taking a aspartate has a carboxylate group, and this asparagine has an amide group. And so this is difference. You're going to take this nitrogen from the glutamine, stick it on the aspartate to make asparagine, and then this, because you took the glutamine nitrogen group, now it has a carboxylate group. And result, you end up with asparagine. So healthy cells can make their own asparagine, but these leukemic cells, they don't make enough of this ASNS. And so they don't make enough of this maker, so they don't make enough asparagine. But normally this is okay because they can get it from the bloodstream, and the bloodstream usually contains like plenty of asparagine, that this isn't a problem. But if you were to deplete the blood supply of asparagine, well, now the healthy cells would be okay because they can still make it, but the cancer cells wouldn't be able to make enough to keep up. And because they can't make enough of their own, they'll actually start like shipping out what they do have And so then they get even more depleted, and then they don't have enough protein in order to do all of the protein making stuff that they need to do. And they need a lot of it because as cancer cells, they're growing and dividing, and they need a lot of nutrients in order to do so. And so if you deplete the blood supply of the asparagine, then these cells can't survive. And they go through like autophagy or like activated, like they program cell death. So not good stuff for the cancer cells, but the healthy cells are okay because they can make their own. More on why this is, there can be side effects and stuff though later in the post. But so the basic idea is that you're adding asparaginase. It's going to affect the leukemic cells, but not the healthy cells. So how do these enzymes work? Because it's just really cool. So basically asparagine synthetase, this is what the cancer cells don't make much of. This is going to take aspartate and it's going to take ATP. So this is like energy money. This is like a costly thing, energetically costly. And you're going to take the amine group from glutamine, stick it onto aspartate. It's going to give you glutamate, and it's going to give you asparagine. So we've seen like amino transfer reactions before, where we are taking an amino group and sticking it somewhere else. But what's unique about this is that we're replacing on the end of the aspartate, we're adding a second amino group. So when we've talked about like transamination reactions in the past, we're typically talking about replacing this alpha amino group. So the one that's like attached to the R group and like this main amino acid group thing. But here we're talking about adding on to the end of the side chain. So it's a little different than what we've seen before, and it's going to use a different mechanism. It's got to be a little trickier. And so this really cool enzyme, asparagine synthetase, basically what's going to happen is it's going to take ATP, and it's going to be like this two-part reaction that's happening like in the same enzyme. It's really cool. So at one site, the aspartate is going to get activated. So basically you need to provide some energy to make this favorable to happen. And so this is going to be done with ATP. So this aspartate, it can basically like this oxygen can attack this alpha phosphate group, so the one closest to the ATP, and now it's going to get stuck on there, and you get this beta aspartyl AMP intermediate. And then you lose this these two phosphate groups, and then this could get further hydrolyzed to give you a boost of energy, because these phosphate groups are like clamp springs, and so you have this negative charge like all having to be held tightly together, and if you break it, then you release energy. And this makes this favorable, and it also makes it so that you're not just going to like go backwards. But at this point, now what's going to happen is you need to replace this AMP with a nitrogen group, and you need a source of that nitrogen group, and that nitrogen group is going to come from glutamine. So glutamine is like asparagine, but it has another carbon in its chain. And so we're trying to make asparagine. We don't have asparagine. We have aspartate. We have glutamine, but glutamine's too long. Aspartate doesn't have the amino group, so let's do a swap. And so we're going to take this amino group from glutamate, and so we need to release it. We need to cut it off, and we're going to cut it off with water, so it's going to get deamidated. So we're taking this amino group off with this hydrolysis, and then this is going to release this ammonia that's going to travel through the protein. So it's all happening. So normally you don't want to just like release ammonia, but it's okay here because it's going to travel through the enzyme, and then it's going to attack this carbonyl carbon, kick off this AMP, and now you get asparagine. Yay! Okay, so we can make asparagine, and this is the thing that the cancer cells aren't very good at making. And so it's going to basically, it has this catalytic threonine. So it has this threonine, which has this OH group, which can act as like a nucleophile. More on this in another post, but basically it can attack this carbonyl carbon. So this oxygen is pulling electrons away from this carbon. This oxygen, so this carbon's partly positive.
This oxygen is nucleophilic. It likes things that are partly positive. It's going to attack that, kick off this amino group as ammonia. But when you get this intermediate where you have this asparagine is going to be stuck on the asparaginase and this like covalent linkage, this beta acyl enzyme intermediate. So we saw an intermediate up here where we're attached to AMP, here we're attached to the enzyme. And then this water can come and break this back off. So you end up with this enzyme regenerated so it can do it again to another molecule of asparagine. So you get aspartate and then the ammonia released. And so because you've broken it down and you've regenerated the enzymes, so that's what the cool thing about enzymes is that they're like the Energizer bunny, they never stop. They can speed up or catalyze a reaction without getting used up in the process. And so they can do it again and again and again. And so it can keep depleting the blood. So even if the cancer cells like start shipping out whatever they do have, kind of like help replenish the stock's just going to keep getting depleted. And so this is why you have, it'll have problems, but the healthy cells will be okay because they can keep making more. But the treatment isn't perfect because although you can be messing up a lot of more of the metabolism than just the asparagine levels. So basically, asparaginase prefers to break down asparagine, but it can also break down glutamine. And so now you're messing with multiple amino acids. And basically, because all of the different pathways, all the different metabolic pathways, so all the different making and breaking of molecules in your body, it's all like an interconnected web. And so if you mess with something, there's like other things that can get messed up. And so you get these global changes in metabolism that can cause problems. Even if the healthy cells can make their own asparagine, now you can be causing problems like altering glycolysis. So altering whether you're breaking down sugar or whether you're breaking down fat. There's a bunch of problems that can arise. So it's not like a perfect treatment, and there are side effects. The asparaginase that's typically used is purified from E. coli or Escherichia coli. So it's this, there's a lot of really safe strains of E. coli that are used a lot in the lab. And one of the things that they apparently have a really good asparaginase, and you can grow E. coli really easily and stuff. And so they can purify out this asparaginase from E. coli and use that as a treatment. Problem is some people develop like a hypersensitivity reaction. So they have like an immune reaction to it because you're sticking in like this foreign protein into these people. So an alternative, if that happens, if a patient has a reaction to it, is that they can give them like a version from a different bacteria, Erwinia chrysanthemi. And so these are different bacteria, so they have a slightly different asparaginase protein. And so this can, they can have, like they might not have a reaction to that one. And so this can be used as like a second line treatment. So another thing is that, so originally they were giving this in just like native form. So basically unmodified, just like purify it from the bacteria. And this is called like Elspar. But now it's given typically as like in its pegylated form. And so this is available as Oncaspar. And then Erwinia is Erwinase. I don't know. Sorry. But yeah, check out this review article and others if you're really interested. But basically these versions nowadays, they're pegylated. So they have this polyethylene glycol. So this it's like a chain of carbon and oxygen repeating units, and this is used commonly in a lot of pharmaceutical products. Basically, it is going to, it does things like increase the solubility and kind of like hide it from the immune system. So this can have increase the stability, various effects, and so this is typically given in its pegylated form. And it's typically given as part of like a combination therapy rather than just alone. So a bit about the history of the treatment. So it goes back to the 1950s. So this is from my blog. There's more on there too if you want to know more. But basically in 1953, Kidd found that guinea pig serum caused regression of implanted lymphoma cells in mice. So basically, when they had mice and they put lymphoma cells in the mice, and then they treated it with guinea pig serum, the guinea pigs, there was an improvement. And so now they had to try to figure out what it was in the guinea pig serum that was causing this. And so this is another scientist, Broom. He found that the anti-lymphoma effect was coming from asparaginase that was in the serum. Then Jollowby et al. And Hill et al. Tried this asparaginase treatment in human patients in the mid-1960s. But it was hard because they needed a source of a lot of asparaginase. And so this treatment didn't really take off as this treatment didn't really take off as a thing until the 1970s when they were able to produce this asparaginase at larger scale, so they had enough to do like big trials. And those big trials helped, and so now it's become a standard part of ALL treatment. It's on the WHO's list of essential medicines. It's also used for treating some forms of acute myeloblastic leukemia or AML. So now let's talk a little bit more about asparagine. So this is asparagine. So it's one of our protein letters, so our amino acids. And in its, like all amino acids, it has an amine group, a carboxyl group, and a unique side chain or R group. And so in its side chain, it has 2 carbons and it has this amide. So where you have this carbonyl, so a carbon double bonded to an oxygen, bound to a nitrogen. You might remember amides because they form the peptide bonds. They're the type of bonds that amino acids link through when they form proteins. So you get a bunch of amino acids, link them up through peptide bonds. So through these amide bonds, and then those fold up to make a protein, and they fold up based on the unique properties of the various R groups or side chains of the different amino acids. And so in the case of asparagine, so its abbreviation is ASN or N, its side chain, this is its side chain, and it has another amide in this chain. So it looks, you might think, oh, it looks similar to what we talked about yesterday if you've been listening. And so yes, it looks similar to aspartate. And we'll see that you can interconvert between the two of them. So aspartate has our carboxylate group on its end. And when we're talking about asparagine, it has an amide on its end. So you might also be kind of reminded of lysine, but it acts very different than lysine. So both of them have nitrogens in their side chain. They end in non-nitrogen. But in addition to having a longer carbon chain beforehand, the lysine has a longer carbon chain. It also is usually positively charged. And because it doesn't have an amide, it just has this primary amino group. So it just has this nitrogen attached to a carbon and then attached to protons. And so it can act as a base, take a proton, and it usually does. And we usually find it in this protonated state in our body. When you have an amide, however, this is going to be resonance stabilized. So just like we see resonance stabilization when we're talking about the backbone of proteins. So just like the backbone of proteins is resonance stabilized, meaning that the electrons are shared throughout the oxygen, carbon, nitrogen. So you have that sharing of the electrons, this resonance or electron delocalization. That's going to be happening in the amide at the end of the asparagine. And so this is going to make this nitrogen. So it's going to have another hydrogen because it's attached in the peptide bond, it would be attached to a carbon, but in asparagine, it's going to be attached to another hydrogen, but it's only attached to one other hydrogen because it's not acting as a base and taking another proton, because it is using those electrons to actually delocalize, and it's sharing those electrons. So you can think of the double bonded character between the oxygen and nitrogen, both having double bonded character, and that's involving the sharing of the lone pair. So that lone pair isn't going to go take up a proton. And this is why you're not going to see asparagine is not going to be like positively charged. It can, however, sometimes act as a nucleophile if it gets deprotonated and then it has a lone pair free. And what can happen then is that you can actually get it post-translationally modified. So translation was the process when we linked all the amino acids together, and then we can also make changes to them after they're linked. And so we call these changes post-translational modifications. And there are different types of post-translational modifications that you can do to different amino acids. And with asparagine, you can attach sugars to them to make glycoproteins or proteoglycans. Or so a glycoprotein is basically where you have more of the protein, less of the sugar, and the proteoglycan is where you have a lot of sugar and less of the protein. And then peptidoglycans are just like have short peptides attached to sugar. But basically, you can link sugars up to make these glycoproteins. You can link them through asparagine, and we call this N-glycans, when you attach to an asparagine residue. And so you can see that this nitrogen, it's going to attach to this sugar chain. And so yeah, I'll fill these like circles and squares, and diamonds look weird. See the post on glycobiology. Basically, those are all different. These are just abbreviations for different sugar units. So asparagine is hydrophilic, so water likes hanging out with it. It has this amide group. It has this polarity, which is going, so basically it has like partial charged regions. It can also form hydrogen bonds. So this makes water really happy. So when we talked about hydrogen bonding, we talked about hydrogen bonding in the context of like the protein backbone being able to make hydrogen bonds. But you can also remember that with asparagine, basically we have a protein backbone in like a peptide. We have that like a amide in the end of the amino acid, not just in the backbone. And because we have this, then we can form these hydrogen bonds to things. And so because it can form these hydrogen bonds and stuff, water likes forming hydrogen bonds. And so water will can form hydrogen bonds with the asparagine and make it really happy. And we'll talk more about this sort of thing tomorrow when we're talking about glutamine, which is like asparagine, but it has one longer chain. So glutamine is longer than asparagine, this is going to make it a little more like flexible. Asparagine is glucogenic. Basically, we can make sugar from it. How we can make glucose from it is we could convert it to oxaloacetate, which is like the end product of the tricarboxylic acid cycle, the TCA cycle. So it's called Krebs cycle or citric acid cycle. And so you can make this oxaloacetate from it, and then that can be used to go make glucose. And so we call it glucogenic. Basically, we can take asparagine, and then we can convert it to aspartate, as we had just seen we could do when we were breaking down asparagine with asparaginase. We're converting it to aspartate, removing this amino group. In order to break down aspartate, now we can do this in a way that's similar to how we break down any amino acid. And so basically it involves like transamination. So we can take this amino group. So now we're talking about moving the alpha amino group. So it was harder to, we had to do, use some special machinery to remove this amide amino group from asparagine. But now it's just like any other amino acid. It still has this other amino acid group. And so here we're going to have a transaminase. So basically for almost all of your amino acids, there's at least one transaminase that what it'll do is it'll take the amino group from that amino acid and stick it onto alpha ketoglutarate, which is one of the members of the TCA cycle. And so when it sticks it onto that, you get glutamate and alpha keto acid. So you've removed this amino group. And by now that you have just like everything, all of the amino groups are going to get transferred onto to alpha ketoglutarate to make glutamate. And now you can just focus on getting it off of glutamate in a controlled manner, which can be done with like glutamate dehydrogenase. So you can call this like combination of transamination and oxidative deamination. transamination. So basically here we're just transferring the amine group, and here we're actually like breaking it off. And when you remove the amino group from aspartate, what you are left with is oxaloacetate. So you remove the amide group, amide nitrogen to get aspartate, and then you remove the alpha carbon, the alpha amino group to get oxaloacetate, and then you can use that oxaloacetate to go and make glucose. Yeah, and so that transamination reaction uses, we saw this when we were looking at lysine, where it uses PLP, which is really cool, and it forms this like shift base. So if you're interested in that, check out that post. So its 3-letter code is ASN, and its initial is an N. And so apparently this is because like asparagine, like with an N, so basically A was taken by alanine, and so its N is for this, but that's not a very helpful name because like most of these end with INE. So instead I remember that it's N, remember that it has this amide group with, so it has this nitrogen in its side chain. And that it's not A because alanine has A. And so, yeah, so you kind of have to come up with little tricks to remember kind of the oddballs. And so asparagine is one of the oddballs when it comes to naming. But we should be grateful that they changed the 3-letter code. It had been in old papers and stuff, you'll see it referred to as ASP -NH2. So basically it had aspartate with an NH2, and actually in a lot of the old research, they sometimes had problems telling the two apart because the groups that like amide would get like hydrolyzed off and stuff in some of their procedures, and so they had trouble differentiating them. So sometimes in old papers you'll see like ASP NH2 or whatever, but then that was changed to ASN for asparagine. There are other ones that also have nitrogen in their side chains, and some of these have weird names too, but you just have to remember that if you can remember like some of them, you can kind of narrow things down, and then remember asparagine has the N. And so for example, like glutamine is Q, so like, and that's like glue Q, so you can remember it that way. And so yeah, so remember it's not that, and it's not, but it has this amide bond. If you want to know more about the various names, there's this cool IUBMB article. It's from 1998. I'm Saffron, but it has like histories of the lettering systems and that sort of thing. So pretty cool stuff. And we have to thank for this 1-letter code. We had to thank Margaret Oakley Dayhoff. And she's considered by many to be like the founder of bioinformatics. And she created this she condensed those 3-letter codes to single-letter abbreviations in order to like reduce the amount of data that was needed. And I'm very grateful that she did, because it's a lot easier to look at like the single letters when you have a big protein. So it's easier for computers to handle. It's also easier for once you have memorized or the 1-letter abbreviations, then it is a lot easier to look at like a long string of protein letters and to be able to see what they are and like talk about them instead of having the 3-letter codes, which if you have a really long protein can like totally take up a ton of space. These 1-letter codes a lot when they're talking about like mutations, like or amino acid substitution, because technically the mutation is happening at the DNA level if you want to get pedantic about it, but we typically call them mutations in protein biochemistry. But anyway, so basically we would put like, so abbreviation wise, we put the original letter first and then what it's changed to afterwards, and in between we put like what like number in the chain it was. So a change for my glycine to an alanine would be written as G91A. So these 1-letter codes, when you see them, like all these weird talk about these substitutions, these mutations, these are like the 3-letter codes that we use. And so we use them a lot because we do a lot of site-directed mutagenesis to try to like figure out what various amino acids do in a protein by changing them. More in the blog version if you're interested, but basically as for the asparagus thing, yeah, so it was first found from asparagus. So it was actually, and this was in 1806 by Vacquelin and Robiquet. and they found it from asparagus juice. So this was actually the first amino acid discovery, but they didn't know that it was a protein letter. They weren't looking for protein letters. They were just trying to figure out what was in asparagus juice. They left some out, went on a trip, came back, and there were crystals of it. And so that's how it gets its name. And so it is from, you can find it in asparagine, asparagus, as well as in a lot of other stuff, but it does not give pea the smell. Some researchers apparently were, because they ingested a bunch of it, and then smelled their pea, and it smelled asparagusy. The bad smell actually comes from breakdown of a sulfur compound called asparagusic acid. And people have different versions of the genes for different smell receptors, and so some people can't smell these, but I do, and it smells really bad. But don't blame the asparagine.